Hello and welcome. Today we'll examine one of the most exciting changes in oncology, the move toward precision immunology, cellular therapy, and biologically targeted treatment. What is CAR-T? CAR-T means chimeric antigen receptor T-cell therapy. A patient's T lymphocytes are collected and genetically engineered to recognize a specific tumor antigen. The CAR combines an antigen-binding domain with signaling components such as CD3 zeta and CD28 or 41BB. When the CAR binds its target, the T-cell activates, proliferates, and releases perforin and granzymes. The remarkable concept is that this is a living drug. Engineered cells can expand and sometimes persist in the patient. Where is it working? The strongest results are in B-cell malignancies and multiple myeloma, particularly with CD19 and BCMA-directed CAR-T. Patients with heavily pretreated, refractory disease can achieve profound and sometimes durable remission. This is now a global field, with major programs across North America, Europe, and Asia, particularly China. The breakthrough: solid tumors. Solid tumors have been difficult because of antigen heterogeneity, antigen escape, poor T-cell trafficking, and the immunosuppressive tumor microenvironment. But this barrier has now been breached. In 2026, China approved satricabtagene autoleucel, targeting Claudin 18.2, for advanced gastric and gastroesophageal junction cancer, the first regulatory approval of CAR-T for a solid tumor. Research is expanding into pancreatic, colorectal, lung, ovarian, and brain cancers, using targets including CLDN 18.2, GD2, B7H3, GPC3, and mesothelin. New approaches include dual-target and logic-gated CARs, armored CAR T-cells, and gene editing. Beyond CAR-T, cellular oncology now includes TCR-engineered cells, tumor-infiltrating lymphocytes, NK cell therapies, bispecific antibodies, cancer vaccines, and stem cell approaches. One exciting development is in vivo CAR-T. Instead of manufacturing cells outside the body, researchers attempt to reprogram T-cells inside the patient. Early 2026 clinical data in multiple myeloma showed responses in all 18 reported patients receiving investigational KLN-1010, although these are very early results and require longer follow-up. Medicinal mushrooms. Coriolus versicolor, or turkey tail, and Ganoderma lucidum, or reishi, contain beta-glucans, polysaccharides, and triterpenes that can influence NK cells, macrophages, T cells, inflammatory signaling, and apoptosis. A meta-analysis of 23 randomized trials involving 4,246 cancer patients reported an association between Coriolus/Ganoderma preparations and lower mortality. However, differences between preparations and study quality mean this is promising evidence, not proof that mushroom extracts replace cancer treatment. Ivermectin and fenbendazole. Experimental studies suggest potential anti-cancer effects involving pathways including AKT/mTOR, Wnt/beta-catenin, apoptosis, microtubules, glucose metabolism, and oxidative stress. For clinicians, the key distinction is between laboratory activity, animal evidence, and proven human efficacy. Ivermectin has emerging human observations, but no established Phase 3 evidence for cancer treatment. Fenbendazole remains even less clinically established. These are research hypotheses, not proven cancer therapies. The direction of oncology. The major shift is toward biological precision. Engineering immune cells, identifying tumor-specific targets, activating endogenous immunity, and potentially reprogramming cells inside the patient. CAR-T has shown that a patient's immune system can become a powerful therapeutic weapon. The next challenge is making these approaches safer, accessible, and effective against solid tumors while rigorously testing promising natural compounds and repurposed medicines. The question is increasingly not simply, what drug can kill this cancer, but how can we make the patient's own biology recognize, attack, and remember it? Thank you for joining me. Subscribe and hit the notification bell to stay in touch.